to close in one story which we think could potentially be a clinical application. Now, there are many forms of blindness where the photoreceptors, our light sensors that are in the back of our eye, are gone. And the retina, of course, is a complex structure. Let's zoom in on it here so we can kind of see it in more detail. The photoreceptor cells are shown here at the top, and then the signals that are detected by the photoreceptors are transformed through various computations until finally that layer of cells at the bottom, the ganglion cells, relay the information to the brain, where we see that as perception. In many forms of blindness, like retinitis pigmentosa or macular degeneration, the photoreceptor cells have atrophied or been destroyed. Now, how could you repair this? It's not even clear that a drug could cause this to be restored because there's nothing for the drug to bind to. On the other hand, light can still get into the eye, right? The eye is still transparent, and you can get light in. So what if we could just take these channelrhodopsins and other molecules and install them on some of these other spared cells and convert them into little cameras? And because there's so many of these cells in the eye, potentially they could be very high-resolution cameras. So this is some work that we're doing. It's being both led by one of our collaborators, Alan Horsager at USC, and being sought to be commercialized by a startup company, EOS Neuroscience, which is funded by the NIH. And what you see here is a mouse trying to solve a maze. It's a 6-arm maze, and there's a bit of water in the maze to motivate the mouse to move, or he'll just sit there. And the goal, of course, of this maze is to get out of the water and go to a little platform that's under the lit top port. Now, mice are smart, so this mouse solves the maze eventually, but he does a brute force search. He's swimming down every avenue until he finally gets to the platform. So he's not using vision to do it. These different mice are different mutations that recapitulate different kinds of blindness that affect humans. And so we're being careful in trying to look at these different models so we come up with a generalized approach. So how are we going to solve this? Well, we're going to do exactly what we outlined in the previous slide. We're going to take these blue light photosensors and install them onto a layer of cells in the middle of the retina, in the back of the eye, and convert them into a camera. Just like installing solar cells all over those neurons to make them light sensitive. Light is converted to electricity on them. So this mouse was blind a couple weeks before this experiment and received 1 dose of this photosensitive molecule in a virus, and now you can see the animal can indeed avoid walls and go to this little platform and make cognitive use of its eyes again. And to point out the power of this, these animals are able to get to that platform just as fast as animals have seen their entire life. So this preclinical study, I think, bodes hope for the kinds of things we're hoping to do in the future.